Hello teachers, hello students. In lesson 1, we have seen that about photosynthesis. Also, in lesson 2, we will see about the second part of photosynthesis. In lesson 1, we have seen the light-dependent photosynthesis. In the light-dependent photosynthesis, NADPH and ATP is produced in the light-dependent photosynthesis. Also, oxygen is released to the atmosphere. In the lesson 2, we will see that the photosynthesis, which is not light-dependent. At the end of this lesson, you will be able to explain light-independent photosynthesis, list the basic stage of Calvin cycle, differentiate between light-dependent and the light-independent photosynthesis, tell the difference of carbon-3, carbon-4, and the complantis. Also, you have to locate the site of photorespiration. Light-independent photosynthesis. This light-independent photosynthesis is called Calvin cycle. Here, this is the second step in the mechanism of photosynthesis. The light-independent photosynthesis is the chemical process of photosynthesis occurring independent of lights, which we call that the dark reaction. This process of photosynthesis or light-independent photosynthesis takes place in the stroma of the chloroplast. This light-independent photosynthesis is purely enzymatic, and it is slower than that of light-dependent photosynthesis. The dark reaction does not require the light energy for the process of photosynthesis. In dark reaction, the sugar are synthesized from carbon dioxide. In the light-dependent one, sugar cannot be synthesized. But in the light-independent photosynthesis, sugar can be synthesized from the carbon dioxide. So, the light-independent photosynthesis use stored chemical energy from the light-dependent reaction to fix this carbon dioxide and create the product that can be converted into glucose. During light-dependent photosynthesis, energy or a chemical energy were produced, and it is stored in the form of ATP and the NADPH. Then this chemical energy, which is produced in the light-dependent stage of photosynthesis, can be used in the light-independent process of photosynthesis to convert or to fix carbon dioxide to produce glucose. Therefore, one of the main products of light-independent photosynthesis is glucose. The energy poor carbon dioxide is fixed by energy storing chemical compound, which is ATP and NADPH2. The process is so-called carbon fixation or carbon assimilation. Here, by this process, carbon-3 plantis can undergo this kind of photosynthesis. Here, carbon-3 plantis are plantis capable of fixing the carbon dioxide into a 3-carbon sugar called phosphoglyceraldehyde. Again, in this process, the enzyme, which is called Rubisco, fix carbon into sugar by using the Calvin cycle. Here, this fixation of carbon dioxide by the enzyme Rubisco is the first step of Calvin cycle. Wheat, rye, oats, and orchards are some of the examples of carbon-3 plantis. Here, carbon-3 plantis exhibit the carbon-3 pathway. That is why they are called carbon-3 plants. It is 3-carbon compound, which is 3-phosphoglyceraldehyde produced at the first steps of Calvin cycle. Here the first carbon compound produced has 3 carbon atoms. Hence the path or the name is so called carbon-3 pathway. The light independent reactions of Calvin cycle can be organized into 3 basic stages. The first stage, which is fixation, and the second step is reduction. Also the third one is regeneration. Let me see these 3 basic stages of Calvin cycles. The first one is, which is carbon fixation. A molecule of carbon dioxide is combined with a carbon acceptor molecules containing a 5-atom compound, which is so called ribulose 1,5-bisphosphate. Now, in the carbon fixation, one molecule of carbon dioxide can be accepted by the ribulose 1,5-bisphosphate. This process is so called a carbon fixation. Then this step gives rise to compound having 6 carbon atoms. This disintegrates into 2 molecules of compound containing 3 carbon called, which is 3-phosphoglyceric acid. Then this reaction is catalyzed by the enzyme which is so called ribulose bisphosphate carboxylase, or simply this enzyme is so called Rubisco. The second step is reduction. It is the second stage of Calvin cycle. Here ATP and the NADPH molecules are utilized to change the 3-phosphoglyceraldehyde molecule into sugar molecules containing 3 carbon atoms. This 3 carbon atom containing molecule is so called glyceraldehyde 3-phosphate. Here this stage has derived its name from the fact that the NADPH donates electrons to 3 carbon intermediates to form glyceraldehyde 3-phosphate. In this case, it donates its electrons. That is why this system is so called reduction, the reduction stage. The third one is regeneration. In this step, some glyceraldehyde 3-phosphate molecules form glucose, while the other need to be recycled so that they can regenerate the ribulose bisphosphate acceptor. Now, this ribulose bisphosphate can be used for the second cycles of Calvin cycle. That is why it should be regenerated. Regeneration needs ATP and involves a complex series of reaction which is called carbohydrate scrambling. In the dark reaction, this carbon dioxide is fixed to carbohydrates, and the carbon dioxide acceptor ribulose diphosphate is regenerated in this case. For this process, or in the Calvin cycle, around 12 NADPH and 18 ATP are required to fix 6 molecules of carbon dioxide into 1 hexose or sugar molecule. In this case, the fructose 6-phosphate can be produced by this process. It would take 6 turns of Calvin cycle to produce 1 molecule of hexose sugar from 6 carbon dioxide molecules. Here, if you see this process of photosynthesis, as we have seen earlier, this glucose or sugar is produced at light independent stage of photosynthesis, but this sugar is produced from 6 molecule of carbon dioxide and 6 molecule of water. Finally, they produces the one molecule of sugar or hexose sugar, in this case glucose. But finally, the oxygen or 6 molecule of oxygen released to the atmosphere as the byproduct of photosynthesis. And this oxygen can be used for the process of respiration in aerobic organisms. Okay. Now, let we see the Kelvin cycle in general. In Kelvin cycle, here there is 6 molecule of carbon dioxide. This 6 molecule of carbon dioxide can be accepted by a chemical compound which is found in the chloroplast. This chemical compound is so-called, which is ribulose bisphosphate. Now, for the process of photosynthesis, 6 molecule of carbon dioxide is important, but each molecule of carbon dioxide enter into the system or to the cycle turn by turn. Therefore, to completely fix the 6 molecule of carbon dioxide by the process of Kelvin cycle, it needs 6 turns of Kelvin cycle. Therefore, when one molecule of carbon dioxide is fixed by ribulose bisphosphate, it forms the first compound. Here, the carbon dioxide molecules are captured by ribulose bisphosphate, resulting in unstable intermediates that immediately break down into 2-phosphoglyceraldehyde. Then this 2-phosphoglyceraldehyde is a 3-carbon atom compound. Now this 3-carbon atom compound is 12 molecule of this phosphoglyceraldehyde is formed. Then after that, ATP is consumed in this stage to convert this phosphoglyceraldehyde phosphate to another compound, which is to the glyceraldehyde 3-phosphate. In the process, also 12 molecule of NADPH is reduced to NADP positive. Here, the phosphoglyceraldehyde is phosphorylated by ATP and reduced by NADPH. Then the removal of the phosphate results in the glyceraldehyde 3-phosphate in this case. As the result, this phosphoglyceraldehyde finally become converted into glyceraldehyde 3-phosphate. Though, through the series of reactions, glyceraldehyde 3-phosphate is rearranged into new ribulose bisphosphate molecules or another sugar. And from this 12 molecule of glyceraldehyde 3-phosphate, 2 molecules of glyceraldehyde 3-phosphate become important to produce a glucose molecules and other carbon compound. Therefore, this is the stage at which glucose or hexose sugar is formed or produced by the process of light-independent photosynthesis. The other remaining 10 molecules of glyceraldehyde 3-phosphate converted into another compound, and it forms 6 molecules of ribulose phosphate. Then after that, here 6 molecule of ATP becomes converted into ADP. As the result, they releases energy, and that energy helps to phosphorylate the compound which is so-called ribulose bisphosphate. Then this is the regenerations of a ribulose bisphosphate. At the beginning, one molecule of glucose become accepted by ribulose bisphosphate, and the ribulose bisphosphate is changed to another compound. But at the end of the stage of Kelvin cycle, this ribulose bisphosphate becomes regenerated. Therefore, the 3 steps or stages of Kelvin cycle is, the first one is, it is the stage of carbon fixation or carbon uptake phase. It is the time at which when one molecule of carbon dioxide become accepted by ribulose bisphosphate. And the second one is carbon reduction phase. It is the stage at which the NADPH is converted into NADPH positive. and converted to another compound which is so-called glyceraldehyde 3-phosphate. And the third one is, it is the regeneration phase. Here, the ribulose bisphosphate, which is the acceptor of carbon dioxide, become regenerated as the third stage of Calvin cycle. Carbon-3 plantes can undergo the process of Calvin cycles, but in carbon-4 plantes, here in some plants like maize, sorghum, and sugarcane, the first product of carbon dioxide fixation is not 3-carbon atom, or not the 3-carbon molecule. Rather, they produces a 4-carbon compound, which is so-called oxaloacetic acid. How this can be produced? Now, in the carbon-3 plantes and in Calvin cycle, the first compound which is produced by carbon fixation is that a carbon-3 atom compound or molecule. But in carbon-4 plantes, the first compound which is produced by the fixations of carbon dioxide is not a carbon-3 compound. It is rather a carbon-4 compound. Therefore, here that carbon-4 compound is so-called oxaloacetic acid. Then the plants that utilize this pathway are commonly called, therefore, a carbon-4 plantes. They are called a carbon-4 plantes because during the carbon dioxide fixation, the first compound which is produced by this process is a carbon-4 atom containing compound is produced. The oxaloacetic acid formed when carbon dioxide is bound to a compound known as phosphoenol pyruvate in the mesophyll cells. In the mesophyll cells of chloroplast, there is a compound which is so-called phosphoenol pyruvate. This phosphoenol pyruvate is the acceptor of carbon dioxide in the carbon-4 plantes. Then, many plantes which live in hot and dry conditions use these mechanisms of photosynthesis, or they follow the pathway of photosynthesis of carbon-4 plantes. These carbon-4 plantes do not have to keep their stomata open as much, and thus they reduce the risk of dying from dehydration. Because most of the carbon-4 plantes live around the hot and the dry conditions. In hot and the dry condition, if they open their stomata, water will move out from their cells, and they may suffer from risk of dying from dehydration. Therefore, to prevent this, they have to close their stomata. At this time, carbon dioxide cannot easily enter into the plant cells or to the mesophyll. So they develop different mechanisms or strategies how to perform their photosynthesis process. Let me see this process, how they can use it. Here, there are two important adaptations for carbon-4 plantes. What are these adaptations? Here, the carbon-4 plantes use an alternate enzyme for the first step of carbon fixation. Here, this enzyme is so-called phosphoenol pyruvate carboxylase enzyme. But in the case of carbon-3 plantes, the enzyme used is the Rubisco. But in this case, the phosphoenol pyruvate carboxylase is used for the carbon fixation, and it has no oxygenous activity. That means they do not fix oxygen from the atmosphere, and has much higher affinity for the carbon dioxide than Rubisco. Here, carbon-4 plantes have specialized leaf anatomy with two different types of photosynthetic cells. What are these two different types of photosynthetic cells for carbon-4 plantes? These are the mesophyll cells. These mesophyll cells are found on the exterior of the leaf and near to the stomata. Near to the stomata. In the case of carbon-3 plants, these mesophiles are found between the upper epidermis and the lower epidermis, and it is found deep in the internal sections of the leaf. But in the case of carbon-4 plants, they are found near to the surface or to the exterior of the leaf. And even if during the dry and hot condition, even if the leaf closes their stomata, this mesophile cell, which is found near to the exterior, has the ability to fix the carbon dioxide by using its enzyme, which is so-called phosphoenol pyruvate carboxylase. As a result, the first compound which is formed by this process is a carbon-4 atom compound. This carbon-4 atom compound then passes to the second cell, which is so-called a bundle sheath of cells. This bundle sheath of cell is the interior of the leaf, which is far away from the stomata. Then, how they can reach on this step? The basic carbon-4 cycle consists of 4 stages. That is, the first stage is fixation of carbon dioxide by the carboxylations of phosphoenol pyruvate in the mesophile cells to form carbon-4 acid. The second one, transport of carbon-4 acids to the bundle sheath, because in the carbon-4 plants uses 2 cells for the process of photosynthesis. Carbon-4 plants uses the mesophile cells and the bundle sheath cells. Now, if the carbon-4 acid is formed in the mesophile cells, then this carbon-4 acid will transport to the bundle sheath, which is far from the stomata. Then decarboxylation of carbon-4 acid within the bundle sheath takes place, and it can generate carbon dioxide inside the bundle sheath, which is then reduced to carbohydrate via the Kelvin cycle. Then once the number of the carbon atom which is found in carbon-4 acid is reduced to carbon-3, then it can enter into the Kelvin cycles. Here, the fourth step is transition of carbon-3 acid, which is the pyruvate or alanine that is formed by the decarboxylation step back to the mesophile cells and regenerations of carbon dioxide acceptor phosphoenol pyruvate. Thus, in carbon-4 plants, carbon-4 carbon fixation has a net added cost of 1 ATP molecule for every carbon dioxide delivered to Rubisco. However, carbon-4 plants are less likely to die of dehydration than carbon-3 plants, because at hot environments or dry condition, they don't need to, or it is not important for them to open their stomata, and they can develop the mechanism through which they can fix carbon dioxide without opening their stomata, because they adapted these strategies. Now let we see this diagram, the carbon-4 photosynthetic pathway. Here we have said that the atmospheric carbon dioxide become fixed, and when atmospheric carbon dioxide fixed, it becomes accepted by the phosphoenol pyruvate. Here, the phosphoenol pyruvate. Here, the phosphoenol pyruvate has 3 carbon atoms. But when one carbon atom becomes added or accepted by this phosphoenol pyruvate, the number of carbon becomes 4. Now, by fixation of this carbon dioxide, the phosphoenol pyruvate, with the help of enzyme phosphoenol carboxylase, changed to the carbon 4 compound, which is oxaloacetate. It's from this process, what we can understand is that the first compound which is produced by this process has 4 carbon atoms. That is why this plant is so-called carbon 4 plantis. In the carbon 4 plantis, in the process of photosynthesis, the first compound which is produced has 4 carbon atoms, which is so-called oxaloacetate. This process will take place in the mesophyll cell, because this carbon 4 plantis undergoes its photosynthesis process in the mesophyll cell as well as in the bundle sheath cells. In the mesophyll cell, the phosphoenol carboxylase helps to fix the phosphoenol pyruvate, one molecule of carbon dioxide, and convert into the 4 carbon atom compound oxaloacetate. Now, this oxaloacetate, by the help of enzyme which is so-called malate dehydrogenase, it converts this compound into malate. Again, this malate is a 4 atom carbon compound. Now, this malate can transport it to another cell in the chloroplast. That cell is so-called bundle sheath cell. Then, when they enter into bundle sheath cell, with the help of enzyme which is so-called malic acid, then the NADP positive reduced to NADPH. At this time, 1 atom of carbon removed from the malate, and it produces carbon dioxide. Here, decarboxylation takes place. In this case, 1 carbon atom is removed from the process. Then a 3 carbon compound is now formed, which is pyruvate. Here, the carbon dioxide, which is produced in the bundle sheath cell, goes to the Kelvin cycle, which is important in the light-independent photosynthesis. Then after, the remaining compound, which is the pyruvate, which has 3 carbon atoms, become enter into or re-enter into that of the mesophyll cell, and by the help of the enzyme which is so-called pyruvate phosphodekinase, it is changes to phosphoenol pyruvate. Here, the phosphoenol pyruvate become regenerated, and now it is ready to accept another molecule of carbon dioxide to proceed the process of photosynthesis. The other point is that CAM, which is Crassulacean acid metabolism plantis. This Crassulacean acid metabolism plantis, or simply CAM, they are the plants which are adapted to dry environment. For example, the cacti, and again, the pineapples are some of the CAM plants which are adapted to dry environment. They use the Crassulacean acid metabolism pathway to minimize photorespiration. This name comes from the family of the plant, the Crassulaceae, in which scientists first discovered the pathway of CAM. Now, this one is the species or the family of Crassulaceae. The CAM plant pathway of photosynthesis first by the scientists discovered by this Crassulaceae family of plants. Here, CAM mechanisms enables plants to improve water use efficiency. The CAM mechanism is similar in many aspects to that of carbon-4 cycle. In carbon-4 plants, the formation of carbon-4 acid in the mesophyll is spatially separated from decarboxylation of carbon-4 acid and from refixations of resulting carbon dioxide by the Kelvin cycles in the bundle sheaths. Now, in the carbon-4, the process takes place in the mesophyll cells and in the bundle sheaths. That is why we can say that it is spatially separated. But in the case of CAM plants, formation of carbon-4 acid is both temporarily and spatially separated. Here in the CAM plants, at night, carbon dioxide is captured by phosphoenol pyruvate carboxylase in the cytosol. That means it is a fluid-filled portions of the cytoplasm. And the malate that forms from the oxaloacetate product is stored in the vacuole. In this case, in the CAM plants, at night the plants capture carbon dioxide by the help of the enzyme which is so-called phosphoenol pyruvate carboxylase in the cytosol. Then the malate that is formed by the process from the oxaloacetate becomes stored in the vacuole during the daytime. The stored malate is transported to the chloroplast and decarboxylated by NADP malic enzyme, and released carbon dioxide is fixed by the Kelvin cycle. During this process, they release the carbon dioxide, but the carbon dioxide which is released by this process become fixed by the Kelvin cycle and enter into the process of light-independent photosynthesis. And the NADPH is used for converting decarboxylated triose-phosphate product to that of the starch. The CAM plants are called, they are succulent, that means they are water storing plants, achieve their high water use efficiently by opening their stomata during the cool. These plants open their stomata during the cool time, that is to minimize the evaporation of water from the plant cell. And again, desert nights, and closing them during the hot and dry day. This is to use their water efficiently. Closing the stomata during the day minimize water loss. But because water and the carbon dioxide share the same diffusion pathway, carbon dioxide must then be taken up at night. Now, the Crassulacean plants or CAM plants take up the carbon dioxide at night time, because at day when they open their stomata, even if carbon dioxide enter into the stomata, but water can evaporate out from the plant cell. So to minimize the process of evaporation or drying out from dehydration, they open their stomata at the night time. So carbon dioxide is incorporated via carboxylations of phosphoenol pyruvate to oxaloacetate, which is then reduced to malate. Here, with onset of day, the stomata close, then preventing loss of water and the further uptake of carbon dioxide. Because the stomata are closed, the internally released carbon dioxide cannot escape from the process. Then from the leaf and then steady is fixed and converted to carbohydrate by the Kelvin cycles. Now, let we see the CAM cycle here. Here in CAM cycle, if this one is in the dark day or in the dark time, the stomata become open in the dark. Then when the stomata open, the atmospheric carbon dioxide become enter into the cell. Then in the cell, it forms acidic environment. Here, carbon dioxide uptake and fixation. Then the leaf become acidified, or leaf acidification. Then this carbon dioxide which enter into the cell, and the cytosol, it combines with the phosphoenol pyruvate and forms the 4-carbon atom compound, which is oxaloacetate, by the process or by the help of the enzyme which is so-called phosphoenol carboxylase. Then the NAD positive malic dehydrogenase reduces NAD H to that of NAD positive, and in the process the compound which is so-called malate is formed. Then this malate changed into the malic acid and finally enter or stored in the vacuole. During the light time or the daytime, the stomata become closed. When the stomata is closed, here prevents water loss and the carbon dioxide uptake. And again, here the decarboxylated carbon dioxide inside the cell, here decarboxylation is stored malate and the fixation of the internal carbon dioxide deacidified. That means in the process from this malic acid, 1 carbon removed by the process of decarboxylation. This results in the deacidifications of the leaf cell. Then now in the light time, the malate which is stored in the vacuole now move out to the cytosol, and it is converted into the compound which is so-called malate. Now this malate become enter into the chloroplasts, which is the site at which photosynthesis takes place. Now when malates enter into the chloroplast, the enzyme NAD positive malic enzyme can catabolize the malate and remove 1 molecule of carbon dioxide. As a result, 1 atom carbon dioxide is removed. The process is so-called decarboxylation. And the other remaining compound, which is so-called pyruvate, is formed and it is changed into starch. But in this process, the carbon dioxide which is removed or released as a result of decarboxylation can be used by the Calvin cycle for the process of photosynthesis. This is how the CAM cycle can be completed to proceed the process of photosynthesis. This is all about photosynthesis. In photosynthesis, we have seen both light-dependent and the light-independent photosynthesis. Here in lesson 2, we have seen that light-independent photosynthesis. In the light-independent photosynthesis, the glucose or hexose sugar is formed. In the Calvin cycle, the carbon-3 plantes can fix the carbon dioxide, and the first compound which is produced by carbon-3 plantes, so-called that a carbon-3 compound which is formed in the process, but in the carbon-4 plantes, carbon-4 atom containing compound is formed, but in the CAM plantes, again the carbon-4 containing compound is formed. Here, in this way, the CAM plantes have different sites at which the process of carboxylation takes place. For example, once the atmospheric carbon dioxide enters into the cytosol and it's changed into the oxaloacetate, then finally it is changed into the malic acid. This malic acid can be entered into the vacuole, and from the vacuole at the daytime the malic acid become converted into malate in the cytosol, and this malate can be decarboxylated and release carbon dioxide. The carbon dioxide released by this process can be taken by the Calvin cycle, and this carbon dioxide can be used for the process of light-independent photosynthesis. Photorespiration is the process which involves oxidation of organic compounds in plants by oxygen in the presence of light. in the photorespiration. What is fixed is oxygen, not carbon dioxide. And the process of photosynthesis, the enzyme Rubisco fixes carbon dioxide for the process of photosynthesis, but in the photorespiration, Rubisco fixes oxygen because it has oxygenous activity. Therefore, photorespiration is the process which involves oxygenation of organic compounds in plants by oxygen in the presence of light. Like ordinary respiration, this process also releases carbon from organic compounds in the form of carbon dioxide, can be released by the process of photorespiration, but does not produce ATP. Therefore, in the process of photorespiration, no production of ATP. And again, the photorespiratory substrate is so-called the glycolate. The glycolate. Here, Rubisco, which is the enzyme used to fix carbon dioxide in the process of photosynthesis, instead of combining with carbon dioxide, it combines with oxygen in the case of photorespiration. This type of oxidation of oxygen to ribulose bisphosphate molecule is known as, that is, the photosynthetic co-oxidation cycle, or it is called glycolate pathway, or it is called carbon to photorespiratory carbon oxidation cycle. Therefore, this process can be called by these three names. Here, photorespiration is common in carbon-3 plants, but highly reduced in carbon-4 plants, and again totally absent in CAM plants. Therefore, photorespiration is common in carbon-3 but absent in CAM plants. Here, the enzyme Rubisco can use either carbon dioxide or oxygen as the substrate. It has these abilities because it has the ability to fix carbon dioxide, as well as it has oxygenous activity. So it has also the ability to fix oxygen. Now, when it fixes oxygen in the process will be photorespiration. The enzyme Rubisco, at which condition can it fix carbon dioxide or oxygen? If you see that, here at low temperature, and again high ratio of carbon dioxide and oxygen, Rubisco fixes carbon dioxide. And the carbon dioxide which is fixed goes to the Kelvin cycle, and it enters into the process of light-independent photosynthesis. But when there is high temperature, and again low carbon dioxide oxygen ratio, the Rubisco fixes oxygen, and the process enters into the photorespiration, because Rubisco has the affinity to fix both carbon dioxide and oxygen. But it fixes carbon dioxide when there is low temperature and high carbon dioxide oxygen ratio, and it fixes oxygen when temperature is high and low carbon dioxide oxygen ratio. Now, photorespiration begins in the chloroplast, then the Rubisco attach oxygen to ribulose bisphosphate in the oxygenous reaction, in this case. Here, 3 carbon compound, which is phosphoglyceraldehyde, and 2 carbon compound, the phosphoglycolate, can be produced by the enzyme Rubisco. Here, the 3 phosphoglyceraldehyde is a normal intermediate of the Kelvin cycle. But the phosphoglycolate cannot enter into the Kelvin cycle. So it is 2 carbon are removed then or stolen from the cycle or from the Kelvin cycle. If Rubisco fixed oxygen, they produces two products. One is the 3-phosphoglyceraldehyde. The other one is phosphoglycolate. This 3-phosphoglyceraldehyde is a normal intermediate for the Calvin cycle. It can directly enter into the Calvin cycle. But the second product, which is phosphoglycolate, is a 2-carbon compound. But this cannot enter into the Calvin cycle. As a result, the 2-carbon can be removed from the cycle, or it can be stolen from the cycle. Then, at the end, 3/4 of the carbon that enters the glycolate pathway, that means the photorespiratory pathway, as a phosphoglycolate can be recovered, while only 1/4 is lost as the carbon dioxide. Let me generally summarize this process in the photorespiratory cycles. In photorespiration, here the enzyme Rubisco has the affinity to fix both carbon dioxide and oxygen. When the Rubisco fixes oxygen, it produces two products. One of the product is 3-phosphoglyceraldehyde. The other is the phosphoglycolate. Here, the phosphoglyceraldehyde is the normal intermediate for the Calvin cycle. Therefore, it enters into the Calvin cycle. But the other one, which is the glycolate, is the 2-carbon compound, is not the intermediate of Calvin cycle. Therefore, it is removed from the chloroplast. In the first case, this process takes place in the chloroplast. Now, the glycolate removed from the chloroplast, then it enters into another organelle which is so-called peroxisome. In the peroxisome, this glycolate can become changed into glycine. Then the glycine from the peroxisome become transported into the mitochondria. Here in the mitochondria, the glycine become changed into 1 molecule of carbon dioxide. In this case, the carbon dioxide become released. This is the decarboxylation process takes place, and another compound serine is produced. Now, here the serine, which is produced, enter into the peroxisome. Then in the peroxisome, this serine converted into the glycerate. But as we have said earlier, this glycerate is the normal intermediate of Calvin cycle. Then it enters into the Calvin cycle. Therefore, in the process of photorespiration, 3 organelles participate. The first one is chloroplast. In the chloroplast, oxygen is captured or fixed by Rubisco, and it's the product 3-phosphoglyceraldehyde plus phosphoglycolate is formed. But the 3-phosphoglyceraldehyde enter into the Calvin cycle. The remaining product, which is phosphoglycolate, changed into glycolate, and it is transported to another organelle which is so-called peroxisome. Now, in the peroxisome, the glycolate changed to a compound glycine. Then the glycine enter to the third organelle, which is so-called mitochondria. In the mitochondria, the glycine become decarboxylated. That means it removes 1 molecule of carbon dioxide. In this case, this carbon dioxide removed from the system, and another compound is formed in addition, which is serine. This serine re-enter into the peroxisome. In the peroxisome, the serine converted into glycerate. Now, this glycerate re-enter into the chloroplast. Then the glycerate is, since it is the normal intermediate of Calvin cycle, it enter into the Calvin cycle and enter to the system of photosynthesis that produces or synthesizes organic compounds or glucose without the use of light energy. This is how photorespiration takes place. To revise the process of photorespiration, here the organelles participates are the chloroplast. Inside the chloroplast, two products are formed, the phosphoglycolate and that of the phosphoglycerate. Then the phosphoglycolate enter into the second organelle, which is so-called peroxisome. And from the peroxisome, again, it is transported into that of the mitochondria. In the mitochondria, the carbon dioxide released or removed out. That's the process of decarboxylation takes place. Then after that, the serine re-enter into that of the peroxisome. From the peroxisome, then again, it enters into the chloroplast and it enters into that of the Calvin cycle. This is how photorespiration takes place. This is the end of lesson 2. Thank you very much.